One of the top concerns for Hoosiers is, are the vaccines protective against the new COVID-19 variants? Well, here to talk about this today is Dr. Eric Barker. He is the Dean of the College of Pharmacy at Purdue University. Welcome.
Thank you.
What do you think about this? We have these new variants, they're spanning the US. Our people are concerned. Are these vaccines, Pfizer, Moderna, Johnson & Johnson, protective against these variants, or do we not know yet?
Well, the jury is still out. We're getting reports both in human clinical trials as well as laboratory settings that are giving us some clues. It does appear that for some of the variants, the vaccines and the immunity that the vaccines generate may be somewhat less effective, but it's important to keep a perspective on this because even a less effective immune response can be a sufficient immune response to prevent severe disease. And at the end of the day, that's one of the things we care most about is keeping people out of the hospital and having those severe cases of COVID-19.
And I understand at Purdue, you're doing something very specific to test these variants. Can you tell us what that is?
So early in January, our animal disease diagnostic laboratory was monitoring the nucleic acid tests that we do here on campus. That's the PCR test that's used as a diagnostic test for COVID-19. And what they were picking up was something that had been learned over in the United Kingdom with the UK B117 variant that had become dominant there. And that is that one of the genetic markers actually disappears from the diagnostic test. It's known as an S gene or an S gene dropout. They began to identify that very early in January and then allowed us to do whole genome virus sequencing to actually look at what variants we truly were seeing. And so early in January, we did identify a couple of the B117 variants. These were particularly from arriving students from international locations. We were able to put them in quarantine immediately. They were not circulating in the general population. So we were able to isolate them and prevent further spread. But really, with the work of the Animal Disease Diagnostic Laboratory in the College of Veterinary Medicine, we were able to identify and then sequence the variants. And now we're doing some random sequencing on positive cases to get a clearer picture of what kind of variations and variants we might be seeing on campus. And the data is still out on that.
That's so interesting. I mean, I understand that Purdue University is one of the first labs to be doing this.
Again, very pleased and proud of the work from the Animal Disease Diagnostic Laboratory. These are scientists that have been studying infectious disease in the furry patients and the animals for many, many years. And they're just now translating what they know from animal disease to human disease and giving us that opportunity. And we're really pleased to be able to be one of the leaders in the state doing this variant analysis.
Well, that's so interesting, Dean Barker. Yes, and congratulations. So innovative work that you guys are doing. And I want to circle back to something. For those of us who don't know what sequencing is, can you explain that for us?
So whole genome sequencing is an approach where you actually take apart the genetic sequence of a virus. You can do this for whole organisms, mammals, really any organism. And several years ago, we would have recalled that the human genome was fully sequenced. But now we have much more rapid analysis approaches for doing genetic sequencing. And we have that capability here on campus. And essentially what it does is it tells us every single what's known as a nucleotide, which is essentially a part of the genetic code. It breaks apart the genetic code one bit at a time and reports that out. So then we can see the entire genetic sequence for an organism. In this case, it's the COVID-19 virus.
And when we talk about this genetic sequencing, and I understand, of course, there's specifics, but how different are these variants really? I know we have 5 from what I understand so far.
Right. So really what we know is in any time there is a virus, particularly in this strain of viruses that the coronaviruses fall in, which are known as RNA viruses, when they replicate inside a human cell, they will make mistakes. There will be errors that are made, and those errors lead to mutations in that genetic sequence. So that's a normal thing that occurs in any virus. It's why seasonally we have different flu strains from one season to the next, because there are mutations that occur in the flu virus, the same as occurring in these coronaviruses. And so if you think about the sequence of that gene, the genome for coronavirus, it's about 30,000 pieces of information that are strung together. When you are replicating that, even if there's one error or two errors for every 1,000 or 5,000, that means there will be some level of mistakes occurring. Many of those mutations are lethal to the virus. The virus can't survive, but some of those mutations are actually favorable and they get propagated. And that's what we see with any viral disease. I've had to encourage folks to get calibrated a little bit, because we've had mutations of the virus from the very beginning. And now we're just seeing that over time, some particular mutations become favorable and they become the predominant strains that we see.
Well, Dean Barker, I think that's a really important point that you make, that this has been mutating since the very beginning, and these strains, yet they're making headlines and seem to be different than previous strains. But this has been happening since the very beginning, and that really puts it into context. So tell us, anything else that you'd like to say about the work that you're doing, and when can we expect results?
I would anticipate over the next 2 to 4 weeks, we'll be getting a lot more sequencing data on the whole virus genomes that we're sequencing here on Purdue's campus. We'll get a better sense of the landscape of what variations are out there and what that really begins to look like. Our concern really becomes when those variants do two things. One is the case with many of the current variants that are becoming more predominant, the UK, the South African, the Brazilian variants that are more transmissible. The spike protein on the surface of the virus, it's essentially the hand that reaches out to connect with our human cells. That hand is actually connecting more tightly with human cells, which allows it to infect more abundantly. And that transmissibility actually becomes of concern. But more importantly, if there's anything in the virus, in these mutant forms of the virus that lead to more severe forms of the disease, which so far we're not really seeing. And that's the thing we'll have to keep our eyes on.
Right. And I want to ask you, if I was in your laboratory, what would I be seeing? Would I be seeing Petri dishes? Would I be seeing vials of plasma? Or what would I be seeing? And what would you be doing with these instruments, tools, and things that are available for you to actually test this?
So the samples that are collected for the diagnostic samples are pretty much what you would have seen if you've had any COVID test done. That is a swab in a test tube. And those then get taken to the lab, and those become our feeder samples. And so we would draw nucleic acid material from those. And then we have large pieces of equipment that are automated sequencers that will again, dissect that genetic code bit by bit. So what you would see is some large boxes of equipment, some test tubes, and a whole lot of folks and people that are moving fluids and liquids from one tube to the next. And that's really how those experiments are done.
It's so interesting. I mean, I wish I could be in your lab at some point, maybe a fly on the wall there. But and then so we have these antibodies in a test tube, in a vial, and they're swabs, and they're swabs of antibodies. Is that correct?
They're
actually,
is that what I heard? Yeah, so those are actually the swabs from the nose that actually are not the antibodies. Those are the actual viral samples that we've collected. And so we're sequencing the actual virus, and that's the sample that we would normally do for diagnostic purposes.
Okay, and then from there, take me through again. You have this sample of the virus,
Yep.
and then what is added to that? What do we add to that to figure something out?
So the tube that we have with the swab, we would extract some of the buffer, the fluid that the swab is sitting in, and then we would add chemical reagents to that to isolate the nucleic acid material, the viral nucleic acid material. And then a sequence of tubes would be laid out where reagents, our buffers, our chemical reagents are added to those tubes that then prep them, that then go into the DNA sequencing analyzer so that we can get that genetic material and that genetic sequence.
Wow, that is so interesting. Well, Dean Barker, it was so great to great to have you here. And thank you so much. And congratulations on all this work that you've been doing and now and in the past. Thank you.
Thank you. It's my pleasure.